Nature has given us compounds, salicylates, that make the active ingredient in aspirin. But instead of coming in a synthetic pill, they're embedded in foods that nourish the body in multiple ways. So these compounds not only work in the bloodstream but also influence the microbiome, regulate inflammation, and improve vascular health at the cellular level. And the beauty is that they do this gently, without the collateral damage that aspirin sometimes causes. Let's start with one of the most well-known natural sources of salicylates: berries, especially blackberries, blueberries, strawberries, and raspberries. These fruits are rich in natural salicylic acid. But unlike aspirin, they also deliver fiber, polyphenols, and antioxidants. When you eat a cup of fresh berries, you're not just thinning your blood in a safer way. You're feeding your endothelial lining, modulating nitric oxide, improving insulin sensitivity, and strengthening the gut barrier. This is systems biology, multiple pathways enhanced with one food. Now think about herbs and spices. Turmeric, rosemary, oregano, and paprika are all surprisingly rich in natural salicylates. When you flavor your food with these, you are introducing anti-inflammatory agents that work not just on COX enzymes, but also on upstream inflammatory pathways like NF-κB, NLRP3 inflammasomes. Aspirin doesn't do that. These compounds help regulate chronic inflammation throughout the body, from the brain to the joints to the arteries, without eroding your stomach lining. Another food that deserves attention here is olive oil, particularly extra virgin olive oil. It contains uleucanthal, which mimics the anti-inflammatory action of ibuprofen. But in addition, olive oil improves HDL functions, stabilizes LDL particles, and nourishes the endothelium. Combined with its content of polyphenols, it becomes a cardiovascular tonic. Regular use of olive oil in a Mediterranean-style diet has been associated with lower risk of stroke and heart disease, even in populations that don't use aspirin. This isn't coincidence. It's a convergence of biological mechanisms. Grapes, especially red or black grapes, contain salicylates and resveratrol. Resveratrol has been shown to improve mitochondrial function, reduce platelet aggregation, and even mimic caloric restriction at the cellular level. These effects are amplified when grapes are eaten whole, with the skin and seeds. Because the synergy of these plant compounds is where the power lies. You don't get this from a supplement or isolated chemical. You get it from the plant in its full expression. Now let's talk about mushrooms. White button mushrooms and shiitake are among the highest natural sources of salicylates. But mushrooms also bring beta-glucans, which modulate the immune system, and ergothinin, a compound some researchers believe should be classified as a vitamin due to its unique cellular protective properties. When mushrooms are part of your regular diet, you're not just getting the anti-platelet benefit, you're getting DNA protection, improved immunity, and antioxidant capacity. Citrus fruits, especially oranges, tangerines, and lemons, are also natural sources. The white pith under the peel is particularly rich in bioflavonoids and salicylates. When you eat a whole orange, just notch the juice, you get anti-inflammatory benefits that extend beyond what aspirin can do. These compounds help stabilize capillaries, reduce oxidative stress, and even enhance lymphatic drainage. That means better circulation, better detoxification, and better overall function. Even vegetables like broccoli, cauliflower, and spinach contain trace amounts of salicylates. When eaten regularly, they contribute to the body's anti-inflammatory and anticoagulation pathways. But unlike medications, they don't override normal physiology, they support it. That's a key distinction. These foods bring balance, not suppression. Let's be clear, aspirin still has its place in certain acute situations. Heart attacks, post-stent care, and specific high-risk cases. But for most people looking for long-term prevention without the risks of bleeding, these foods offer a compelling alternative. They work gently but powerfully. They don't harm the gut. They don't increase the risk of hemorrhagic stroke. And they offer a host of additional benefits to the immune system, the brain, the liver, and the skin. But this isn't about one superfood. It's about a dietary pattern, a lifestyle of consistent nourishment. When you combine these salicylate-rich foods with proper sleep, daily sunlight, exercise, and stress reduction, the benefits compound. Your inflammation goes down. Your circulation improves. Your risk for chronic disease drops. This is not just good science, it's good sense. It's listening to what nature has provided and aligning our choices with how the body was designed to function. You don't need to pop a pill to protect your heart. You can start with a handful of berries, a drizzle of olive oil, a dash of turmeric, and a plate full of leafy greens. You can build health from the inside out. Aspirin from processed aspirin has long been the gold standard for reducing blood clots and lowering the risk of heart attack and stroke, largely due to its ability to inhibit platelet aggregation through the suppression of cyclooxygenase COX enzymes. This effect prevents platelets from sticking together and forming clots that could block arteries. However, its use is not without risk. One of the most significant concerns, particularly in long-term users, is gastrointestinal bleeding and intracranial hemorrhage. For some individuals, even low-dose aspirin can irritate the stomach lining, leading to ulcers, perforations, or significant blood loss. The medical community has increasingly acknowledged this risk, especially in older populations or those with pre-existing gastrointestinal conditions. As a result, There is a growing interest in alternative sources that can offer similar anti-inflammatory, anti-platelet effects without causing damage to the digestive tract or increasing the risk of dangerous bleeds. In nature, there are plant-derived compounds known as salicylates. These are the very substances from which aspirin was originally developed. Willow bark, the plant source used centuries ago to treat pain and fever, contains salicin, a precursor to salicylic acid. But beyond willow bark, salicylates are found in a wide range of common foods. In contrast to synthetic aspirin, which delivers a concentrated and sometimes harsh pharmacologic dose, the salicylates in whole foods are present in lower concentrations and come packaged with fiber, antioxidants, and phytonutrients. This natural packaging helps buffer the compound's effects, reducing irritation and enhancing its therapeutic profile. Foods like berries, herbs, spices, grapes, citrus fruits, and even certain vegetables contain salicylates in varying amounts. These natural salicylates interact with the same COX pathways as aspirin, but do so in a more moderated fashion. They can reduce inflammation, thin the blood slightly, and improve circulation without the systemic risks associated with long-term pharmaceutical use. Importantly, these natural sources do not disrupt the mucosal lining of the gastrointestinal tract. On the contrary, many of them, like berries and leafy greens, actively support gut health. Their fiber content and prebiotic effects. The human body is highly responsive to these dietary compounds. Once ingested, salicylates are absorbed and metabolized by the liver before entering systemic circulation. At this point, they exert their effects on platelets and vascular tissue, helping to maintain fluidity in the blood and reduce the risk of inappropriate clotting. Additionally, unlike aspirin, which can sometimes excessively suppress platelet function, natural salicylates tend to modulate rather than completely inhibit this system. This allows for a more balanced approach to clot regulation, helping reduce risk while preserving the body's ability to respond to injury. In epidemiological studies, diets high in plant-based foods rich in polyphenols and salicylates have been associated with lower levels of inflammatory markers like C-reactive protein CARP, interleukin-6, IL-6, and tumor necrosis factor alpha, TNF-FT. These compounds also help increase nitric oxide production, which relaxes blood vessels and improves vascular tone. When taken together as part of a diet rich in whole, unprocessed foods, the cumulative effect of these compounds is substantial. Unlike drugs, which often target a single receptor or pathway, whole foods provide a symphony of bioactive molecules that work synergistically to support cardiovascular health. Another key distinction is the interaction with the microbiome. Aspirin, while effective, can damage the mucosal barrier and alter gut flora in ways that may increase susceptibility to inflammation and permeability, also known as leaky gut. Natural salicylate-rich foods, by contrast, promote microbial diversity and contribute to the production of short-chain fatty acids like butyrate, which nourish colon cells and strengthen the gut lining. A well-regulated gut barrier has downstream effects on systemic inflammation and vascular function, creating a more holistic and sustainable method for cardiovascular protection. Moreover, the anti-inflammatory action of natural salicylates extends beyond the vascular system. These compounds help modulate immune responses, reducing chronic low-grade inflammation that contributes to insulin resistance, neurodegeneration, and autoimmune conditions. By targeting multiple inflammatory pathways, including nuclear factor kappa B, NFJB, and the NLRP3 inflammasome, these plant-based compounds provide broader protection than aspirin's more narrow pharmacological mechanism. This makes them ideal candidates for long-term use in disease prevention strategies. It's important to understand that while pharmaceutical aspirin delivers a potent therapeutic effect, its intensity can also be its drawback. The body's hemostatic system, which includes clotting and bleeding responses, is finely balanced. Too much inhibition can lead to uncontrolled bleeding. In contrast, the gradual moderate action of dietary salicylates works in harmony with this system, providing support without overwhelming it. This distinction is critical for populations at higher risk of bleeding, including older adults, people with ulcer histories, or those concurrently taking other blood thinners or NSAIDs. The use of food-based solutions offers a safer, more integrative approach to health, aligning with how the human body evolved to process nutrients and medicinal compounds. Berries stand out as some of the richest dietary sources of natural salicylates, making them powerful tools for supporting cardiovascular health and reducing inflammation without the side effects commonly associated with pharmaceutical aspirin. Blackberries, blueberries, raspberries, and strawberries all contain significant amounts of salicylic acid, the naturally occurring compound that serves as the foundational molecule for synthetic aspirin. When consumed as whole foods, these fruits offer far more than their salicylate content alone. They deliver an array of bioactive compounds including anthocyanins, flavonols, ellagic acid, and fiber, each of which contributes uniquely to improving blood flow, reducing platelet aggregation, and supporting endothelial function. The role of anthocyanins in berries is particularly significant. These deep pigments, responsible for the red, blue, and purple hues of the fruits, have been shown in clinical studies to exert antioxidant and anti-inflammatory effects by scavenging reactive oxygen species and modulating cellular signaling pathways. By reducing oxidative stress, Anthocyanins help protect the delicate lining of blood vessels known as the endothelium. When the endothelium is preserved, nitric oxide levels are better maintained, which in turn allows for improved vasodilation and reduced vascular resistance. This supports smoother, more efficient blood flow and lowers the risk of clot formation. Unlike aspirin, which simply inhibits one aspect of clot formation, berries support multiple aspects of vascular health simultaneously. In addition to anthocyanins, berries are rich in flavonols such as quercetin and kaempferol. These compounds inhibit pro-inflammatory enzymes, reduce lipid peroxidation, and improve mitochondrial function within vascular cells. Through these mechanisms, they contribute to the regulation of blood pressure, glucose metabolism, and inflammatory status, factors that are tightly linked to cardiovascular disease risk. The presence of natural salicylates in these fragments this effect by gently modulating platelet activity without completely suppressing the clotting response. This is an important distinction, as a partial inhibition is often sufficient to reduce cardiovascular risk without tipping the balance toward excessive bleeding. Grapes, especially red and black varieties, offer another compelling example of nature's aspirin-like foods. Beyond their content of salicylic acid, grapes are a potent source of resveratrol, a compound found in the skin of the fruit. Resveratrol has gained significant attention for its ability to activate SART1, a gene associated with longevity and cellular repair. It also enhances mitochondrial biogenesis, reduces oxidative damage, and improves endothelial function. These effects collectively contribute to improved arterial elasticity and reduced platelet stickiness, wearing some of the cardiovascular benefits of aspirin but doing so through a wider biological spectrum. Moreover, resveratrol has been shown to inhibit cyclooxygenase enzymes in a way that complements the action of natural salicylates, further contributing to a reduction of thrombosis and vascular inflammation. Fiber is another major contributor to the cardiovascular benefits of berries and grapes. Soluble fiber in these fruits modulate postprandial glucose spikes, lower LDL cholesterol, and support a healthy gut microbiome. A well-functioning microbiome, in turn, produces short-chain fatty acids that lower systemic inflammation and improve insulin sensitivity. This systemic anti-inflammatory effect further reinforces the body's ability to maintain healthy blood vessels and reduces the likelihood of clot formation. In contrast to aspirin, which may disrupt gut integrity and lead to gastrointestinal bleeding, these fruits actually nourish and reinforce gut health. The seeds of grapes, often discarded or overlooked, are another concentrated source of polyphenols and proanthocyanidins. Grapeseed extract has been studied for its ability to improve circulation, reduce blood pressure, and decrease oxidative damage to lipoproteins in the bloodstream. These compounds complement the salicylate content by promoting vasodilation and reducing vascular stiffness when grapes are consumed whole. Skim pulp and seeds. The synergy of these compounds creates a therapeutic food that works on multiple physiological levels to promote cardiovascular resilience. A regular consumption of berries and grapes has been correlated with reduced markers of inflammation such as C-reactive protein and interleukin-6 in observational and interventional studies. These reductions are not merely biochemical but translate into clinical outcomes such as lower incidence of stroke, myocardial infarction, and metabolic syndrome. Importantly, these outcomes are achieved without increasing of adverse bleeding events, making them particularly suitable for long-term use, especially in populations with elevated bleeding risk or sensitivity to NSAIDs. Even beyond the cardiovascular benefits, berries and grapes exhibit neuroprotective and metabolic advantages that contribute to whole-body health. Their compounds cross the blood-brain barrier and influence brain-derived neurotrophic factor BFDNF, enhance glucose uptake in neurons, and reduce microglial activation. In individuals with insulin resistance or early cognitive decline, these effects are especially valuable. Salicylates in these fruits may play a supporting role by reducing neuroinflammation and improving cerebral blood flow. Taken together, the anti-inflammatory, antioxidant, and vasodilatory properties of these fruits converge to form a dietary intervention that is both powerful and safe rather than relying on synthetic agents that produce a single targeted effect with potential collateral damage. Consuming berries and grapes allows for a more holistic and biologically compatible approach. Their integration into a daily diet supports cardiovascular health not through pharmacologic force, but through nutritional synergy and cellular balance. Herbs and spices like turmeric, rosemary, thyme, oregano, cinnamon, and paprika are among the richest natural sources of salicylates, and their ability to influence inflammation and blood flow extends far beyond the mechanisms of traditional aspirin. These culinary elements have been used for centuries across various cultures not just to enhance flavor, but also for their medicinal properties. Modern science has now confirmed that these foods work on multiple molecular levels, targeting inflammatory pathways, modulating immune responses, and influencing platelet activity in ways that are both powerful and gentle. While aspirin primarily works by inhibiting cyclooxy-1-COX-1 and cyclooxygenase COX-2 enzymes to reduce thromboxane and prostaglandin formation, these herbs and spices go deeper, affecting broader inflammatory cascades and regulating oxidative stress throughout the body. Turmeric, for example, contains curcumin, a bioactive compound with potent anti-inflammatory, antioxidant, and anti-platelet properties. Curcumin does not simply block TEX enzymes like aspirin, it also modulates nuclear factor kappa B NFJB, one of the master regulators of the inflammatory response. NFJB controls the expression of genes involved in cytokine production, immune cell recruitment in chronic inflammatory diseases. By suppressing NFJB activation, curcumin helps reduce the overproduction of interleukins and tumor necrosis factor alpha, contributing to lower systemic inflammation. It also downregulates the expression of adhesion molecules that allow white blood cells to stick to the vascular endothelium, thereby protecting against vascular inflammation and clot formation. This wide-ranging action helps explain why turmeric has shown promise in conditions ranging from arthritis to cardiovascular disease to metabolic syndrome. Oregano and thyme are similarly rich in compounds like rosmarinic acid, thymol, and carvacrol, all of which demonstrate significant anti-inflammatory and antimicrobial properties. Rosmarinic acid is a natural polyphenol that not only exhibits the oxidant effects but also suppresses complement system activation, which plays a key role in inflammatory injury to blood vessels. Thymol and carvacrol have both been shown to inhibit COX enzymes while also reducing reoxygen species that can damage vascular tissues. Importantly, these compounds support the preservation of nitric oxide, a crucial molecule for vascular relaxation and endothelial health. By maintaining nitric oxide bioavailability, oregano and thyme help support pressure regulation and improve overall blood flow dynamics. Rosemary contains its own set of bioactive agents, including carnosic acid and carnosol, which have been studied for their ability to reduce lipid peroxidation and inhibit pro-inflammatory transcription factors. These compounds contribute to the stabilization of atherosclerotic plaques by preventing oxidative modification of LDL cholesterol, thereby reducing the risk of plaque rupture and subsequent clot formation. In addition, rosemary has demonstrated mild anti-platelet activity, which staying away from processed foods, which may help reduce clot risk in a manner that is more balanced and physiological than aspirin's complete inhibition of platelet aggregation. This nuanced action allows for cardiovascular support without significantly increasing bleeding risk. Cinnamon is another spice with multi-layered anti-inflammatory effects. It contains cinnamaldehyde, which not only inhibits KEX activity but also suppresses the release of arachidonic acid, the precursor to both pro-inflammatory prostaglandins and leukotrienes. By working upstream the inflammatory cascade, cinnamon can reduce inflammation at its source. It also improves insulin sensitivity, lowers fasting blood glucose, and reduces triglyceride levels, all of which are important in mitigating cardiovascular risk. The salicylate content in cinnamon contributes to its blood-thinning effects, adding yet another layer of cardiovascular support without the gastrointestinal irritation that aspirin can cause. Paprika and chili powder, rich in capsaicin, also exhibit strong anti-inflammatory actions. Capsaicin acts on the transient receptor potential vanilloid-1 TRPV1 receptor, leading to the desensitization of sensory neurons that release substance P and other neuropeptides involved in pain and inflammation. This mechanism not only reduces inflammation locally, but may have systemic effects on neurogenic inflammation, a contributor to vascular dysfunction. Capsaicin also appears to reduce platelet aggregation and improve blood lipid profiles, including reducing LDL oxidation and enhancing HDL function. These actions further support its role in cardiovascular protection. What makes herbs and spices uniquely powerful is their concentration of polyphenols and their ability to affect multiple physiological systems simultaneously. Unlike synthetic drugs that often target a single receptor or enzyme, these natural compounds influence gene expression, enzyme activity, microbial populations, oxidative stress, and even mitochondrial function. They contribute to lowering blood pressure, stabilizing blood sugar, reducing systemic inflammation, and improving lipid profiles. Without the disruption of the gut lining, a known complication of chronic aspirin use, another key advantage is their bioavailability when consumed regularly in the diet. When herbs and spices are used in cooking, particularly with healthy fats like oil or avocado oil, the absorption of fat-soluble compounds such as curcumin and rosmarinic acid is enhanced. This makes them not only effective but also highly accessible. Integrating them into daily meals provides continuous low-dose exposure to these therapeutic compounds, resulting in cumulative benefits over time. Unlike aspirin, which is often taken in fixed doses and may accumulate adverse effects, the natural salicylates and anti-inflammatory compounds in herbs and spices work in harmony with human physiology, making them ideal for long-term cardiovascular and anti-inflammatory support. Olive oil, particularly extra virgin olive oil, and mushrooms such as white button and shiitake are two often overlooked sources of natural salicylates that offer profound cardiovascular and anti-inflammatory benefits without the well-known side effects associated with aspirin. These foods are unique in that they provide both natural salicylates and a constellation of additional compounds that interact synergistically to enhance vascular health, modulate immune function, and protect against oxidative stress. Their integration into a daily diet does more than just offer an aspirin-like effect. It provides a full spectrum of physiological support that nourishes the endothelium, reduces platelet stickiness, and supports optimal circulation. Extra virgin olive oil is rich in polyphenols, especially oleocanthal, which has been shown to possess strong anti-inflammatory effects comparable in potency to ibuprofen. Oleocanthal acts by inhibiting both CX1 and COX2 enzymes, similar to aspirin, but it does so in a gentler, food-based matrix that avoids damage to the gastrointestinal lining. In addition to its COX inhibitory properties, olive oil contains salicylic acid in modest amounts, contributing to its ability to reduce clot formation and improve blood fluidity. What differentiates olive oil from pharmaceutical agents is its broader range of action. It improves endothelial function, reduces oxidative stress, enhances HDL particle quality, and promotes mitochondrial efficiency in vascular cells. These combined effects create a food that doesn't just mimic aspirin, but surpasses it in terms of systemic health support. Mushrooms, particularly white button and shiitake varieties, contain some of the highest levels of dietary salicylates among non-fruit and non-herb foods. But beyond their salicylate content, mushrooms bring a range of bioactive molecules, include beta-glucans, ergothioneine, and lectins that play important roles in regulating immune function and maintaining vascular integrity. Beta-glucans in mushrooms are known to modulate innate and adaptive immunity, enhancing the function of natural cells and macrophages while simultaneously reducing chronic inflammation. These fibers also help to regulate blood lipids by binding to cholesterol in the digestive tract and reducing its absorption, thereby lowering LDL cholesterol levels and supporting arterial health. Ergothioneine, a powerful antioxidant found almost exclusively in mushrooms and certain bacteria, is another proteetical compound with protective effects against oxidative stress. Ergothioneine is transported into cells via a specific transporter known as OCN1, where it accumulates in mitochondria and other sensitive structures to protect against oxidative damage. This compound helps stabilize endothelial cells, reduces lipid peroxidation, and preserves mitochondrial function in the face of stress, all of which contribute to reduced vascular inflammation and lower risk of plaque formation. The antioxidant activity of ergothioneine also indirectly supports nitric production by reducing oxidative degradation of this key signaling molecule, thereby promoting vascular dilation and blood pressure regulation. The unique nutritional profile of mushrooms also includes selenium B vitamins and vitamin D2, which together support metabolic and cardiovascular health. Selenium plays a crucial role in the activity of glutathione peroxidase, an enzyme that neutralizes hydrogen peroxide and lipid peroxides in the blood. By reducing oxidative stress at the level of the blood vessel wall, selenium contributes to plaque stability and reduces the likelihood of clot formation. Vitamin D2, though less bioavailable than D3, still provides benefits for immune modulation and inflammation control, especially when consumed as part of a diverse whole food diet. Olive oil and mushrooms are also uniquely suited to preserving the gut vascular barrier. Chronic use of NSAIDs like aspirin is known to damage the gastrointestinal lining, leading to increased intestinal permeability or leaky gut, which can trigger systemic inflammation and increase the risk of cardiovascular disease. In contrast, the polyphenols in olive oil have been shown to strengthen tight junctions between intestinal cells, reduce gut-derived endotoxemia, and support a healthier microbiome composition. Similarly, the prebiotic fibers in mushrooms feed beneficial bacteria that produce short-chain fatty acids like butyrate, which help maintain gut barrier integrity and reduce inflammation in the circulatory system. Both olive oil and mushrooms demonstrate mild anti-platelet effects that are clinically relevant, particularly when consumed regularly. While aspirin exerts a strong and irreversible inhibition of platelet aggregation through QX1 inhibition, leading to increased bleeding risk in some individuals, these foods offer a more moderate and reversible modulation of platelet activity. This allows them to reduce the risk of thrombosis while maintaining the body's ability to respond appropriately to injury or surgical procedures. The net effect is a more balanced, sustainable approach to cardiovascular risk management, especially for individuals who are sensitive to pharmaceuticals or at higher risk of bleeding complications. The culinary versatility of both olive oil and mushrooms also makes them easy to incorporate into a daily routine. Whether sautéed, roasted, or added raw to salads, must provide dense nutritional value in a low-calorie, high-fiber package. Olive oil, when used as a dressing or cooking fat, enhances the absorption of fat-soluble nutrients from vegetables and herbs, amplifying their health benefits. This synergistic interaction between olive oil and plant-based food mirrors the complexity and adaptability of human physiology, where health is best maintained through diverse, nutrient-rich, whole food sources rather than isolated pharmacological interventions. Citrus fruits and cruciferous vegetables are foundational elements in a nutrient-rich, anti-inflammatory diet that naturally support cardiovascular health and blood fluidity through their salicylate content and a wide array of complementary phytochemicals. Oranges, lemons, limes, grapefruits, and tangerines contain salicylic acid, particularly concentrated in the white than peel. While many people focus solely on the juice, the true therapeutic potential of citrus lies in consuming the whole fruit, including its fiber and bioflavonoids. These compounds work in synergy to stabilize capillaries, reduce oxidative stress, and enhance lymphatic and vascular circulation. Unlike aspirin, which may compromise the gastrointestinal lining and provoke ulceration or bleeding, citrus bioactives provide gentle modulation of inflammation and platelet activity while simultaneously strengthening connective tissue and promoting detoxification. Citrus fruits are rich in flavonoids such as hesperidin, naringenin, and diasmin, each with documented vasoprotective effects. Hesperidin has been shown to improve endothelial function by increasing nitric oxide availability, reducing vascular resistance, and enhancing capillary tone. Naringenin modulates inflammatory gene expression by downregulating NFJB signaling and reducing levels of interleukin-1 and tumor necrosis factor alpha. These effects are crucial in the context of chronic low-grade inflammation, which underlies atherosclerosis, hypertension, and metabolic syndrome. The combined presence of these flavonoids, along with salicylic acid, means that citrus fruits offer not just a superficial resemblance to aspirin's blood-thinning effect, but a much deeper and broader regulatory influence on vascular health. Pectin, a soluble fiber abundant in citrus fruits, also contributes significantly to cardiovascular function by binding to bile acids and reducing cholesterol absorption in the gut. This leads to lower LDL cholesterol and improved lipid ratios. Pectin also serves as a prebiotic, feeding beneficial gut bacteria that produce short-chain fatty acids like propionate and butyrate, which exert anti-inflammatory effects throughout the body. These metabolites travel through the bloodstream, influencing immune cell behavior, reducing oxidative stress in the endothelium, and enhancing insulin sensitivity, all of which contribute to lower cardiovascular risk. In this way, the salicylate activity of citrus is only one part of a much larger matrix of cardiovascular benefits provided by the whole fruit cruciferous vegetables such as broccoli, cauliflower, Brussels sprouts, cabbage, and kale also contain natural salicylates, but their contribution to vascular and immune health extends well beyond that. They are rich in glucosinolates, sulfur-containing compounds that, when broken down during digestion, produce isothioceanates such as sulforaphane. Sulforaphane activates the NTRF2 pathway, a key regulator of cellular defense mechanisms, including antioxidant production and detoxification enzyme expression. Through this pathway, cruciferous vegetables help lower oxidative damage to blood vessels, reduce chronic inflammation, and improve metabolic flexibility. This biochemical resilience translates into stronger, more adaptable arteries and a lower tendency for clot formation or vascular injury. These vegetables are also powerful sources of vitamin K1, which supports healthy blood clotting by maintaining a balance between coagulation and anticoagulation pathways. Unlike aspirin, which irreversibly inhibits platelet aggregation and can tip the scale toward excessive bleeding, vitamin K helps the body maintain homeostasis. It allows for adequate clot formation in the event of injury while supporting vascular integrity by activating matrix GLA protein, which inhibits calcium buildup in the arterial walls. The result is more elastic arteries and a reduced risk of arterial stiffness and calcification, conditions that strongly predict heart attack and stroke. Fiber in cruciferous vegetables enhances satiety, regulates blood sugar, and binds to toxins and excess hormones in the gut, promoting elimination and reducing systemic inflammation. Additionally, these vegetables support bile metabolism and liver detoxification through their sulfur compounds, enhancing phase detoxification pathways. This reduces the toxic burden on the vascular system and lowers inflammatory mediators in circulation. The presence of salicylates in these vegetables acts in concert with these mechanisms to gently inhibit platelet stickiness and promote smoother blood flow. Indole 3, carbinol, and diindolylmethane DIM, two unique phytonutrients found in cruciferous vegetables, also contribute to immune regulation and hormonal balance. These compounds modulate estrogen metabolism and support the immune system in distinguishing between healthy cells and potentially harmful ones, which is particularly important in the context of chronic inflammatory diseases. They also help downregulate pro-inflammatory cytokines and upregulate protective T regulatory cells, adding another layer of support to the body's defense against chronic vascular inflammation. Both citrus fruits and cruciferous vegetables are water-rich, mineral-dense, and provide vital cofactors such as magnesium, potassium, and folate, which are essential for maintaining healthy blood pressure, red blood cell production, and methylation cycles. These micronutrients enhance the body's ability to repair damaged blood vessels, regulate fluid balance, and produce neurotransmitters that affect stress and vascular tone. Potassium in particular plays a key role in modulating sodium balance and reducing arterial stiffness, further supporting the anti-hypertensive effects of these foods. Their hydrating and alkalizing properties also make them particularly valuable in balancing pH levels and reducing systemic acidity that can otherwise contribute to inflammation and vascular dysfunction. Regular consumption of citrus fruits and cruciferous vegetables has been associated with lower rates of cardiovascular disease, improved endothelial function, reduced markers of inflammation, and enhanced metabolic outcomes. These effects are not due to a single nutrient or count but arise from the synergistic action of natural salicylates, antioxidants, flavonoids, fiber, and essential micronutrients working together to create an environment in the body that is less prone to clot formation, oxidative damage, and chronic disease progress. A salicylate-rich diet does far more than mimic the blunting effects of aspirin. It supports whole body health by gently modulating inflammation, improving circulation, and enhancing physiological resilience through multiple interconnected systems. The body thrives on balance, and natural salicylates found in fruits, vegetables, herbs, spices, and plant oils help maintain that equilibrium without forcing the body into an artificial or oversuppressed state. Unlike synthetic medications that target isolated pathways, whole foods work in concert with human biology, engaging antioxidant defenses, supporting endothelial function, nourishing the gut microbiome, and regulating immune activity in a synchronized manner. This approach respects the body's natural rhythms and fosters internal harmony, which is especially crucial for long-term disease prevention and healthy aging. Dietary salicylates are most effective when integrated into a pattern of eating that emphasizes diversity, color, fiber, and freshness. Rather than isolating a single compound or food, the real power emerges from a daily intake of multiple salicylate-containing foods. Berries in the morning, cruciferous vegetables at lunch, herbs and spices in dinner preparations, and olive oil as a daily fat source. These foods contribute not only their salicylic acid content but also a vast array of bioactive molecules, including flavonoids, polyphenols, terpenes, and sulfur compounds that work together to reduce oxidative stress, stabilize blood sugar, enhance lipid profiles, and promote nitric oxide production for optimal vascular tone. Each of these mechanisms is relevant not only to heart health but also to brain function, immune efficiency, and metabolic stability. The synergy of these foods offers a deeper layer of healing because their compounds often activate similar protective pathways within the body. For example, multiple plant-based nutrients stimulate the NRF2 pathway, which governs antioxidant gene expression, while simultaneously inhibiting NFJB, the transcription factor that triggers chronic inflammation. This dual activation-deactivation response cannot be replicated by aspirin, which primarily works on cyclooxygenase enzymes and leaves other inflammatory drivers untouched. The systemic reach of dietary salicylates means they do not simply thin the blood, but reduce the conditions that make abnormal clotting more likely in the first place. Oxidized LDL particles, damaged endothelium, high-sensitivity C-reactive protein, and elevated homocysteine. The circulatory benefits of these foods also extend to microvascular systems, where capillary health and perfusion are essential for oxygen delivery and waste removal. Natural salicylates help preserve capillary elasticity, reduce red blood cell aggregation, and improve microcirculation in organs such as the kidneys, brain, eyes, and extremities. These effects contribute to sharper cognition, better renal filtration, improved visual acuity, and reduced neuropathic symptoms. Outcomes that are rarely linked to aspirin therapy but are achievable through consistent dietary choices. The ability of salicylate-rich foods to influence macro and microcirculatory systems underlines their role as whole-body regulators. Not just cardiovascular adjuncts, the gut microbiome plays an integral role in the systemic effects of these foods. When a diet is rich in plant-derived salicylates, it also tends to be high in fermentable fibers, resistant starches, and plant polyphenols that serve as substrates for beneficial bacteria. These bacteria, in turn, generate metabolites like short-chain fatty acids and indoles that regulate inflammation at distant sites, including the vascular endothelium, adipose tissue, and central nervous system. Moreover, a balanced microbiome helps maintain tight junction integrity in the intestinal lining, reducing translocation of inflammatory endotoxins into the bloodstream. This gut-mediated protection supports less turbulent blood flow, and lowers the overall inflammatory load. A foundation-reducing factor in reducing clot, enhancing cardiovascular resilience, stress, adaptation, and circadian regulation are additional layers of health influenced by the salicylate-rich diet. Many plant compounds found in these foods have mild adaptogenic effects, helping regulate cortisol rhythms, reduce reactive oxygen species in the adrenal glands, and modulate neurotransmitter balance. This stress buffering capacity is especially important in our modern environment, where chronic psychological and metabolic stress drives sympathetic overactivity, vasoconstriction, and platelet hyperreactivity. By reducing both physiological and emotional stressors, these foods create an internal environment where clot formation becomes less probable because the body remains in a parasympathetic, dominant, repair-oriented state. Physical movement amplifies the benefits of salicylate-containing foods by increasing nitric oxide production, enhancing insulin sensitivity, and improving blood flow to active tissues. When combined with regular physical activity, these foods support efficient oxygen delivery, faster removal of metabolic waste, and a reduced risk of endothelial dysfunction. Their effects on inflammation, oxidative balance, and platelet activity complement the metabolic adaptations of exercise, creating a feedback loop of improved performance and faster recovery. Together, movement and a salicylate-rich diet form a natural strategy for maintaining arterial health without the risks associated with synthetic inflammatory medications. Sleep quality and light exposure also play into the equation. Natural salicylate foods support melanin production through their influence on tryptophan metabolism and oxidative stress reduction. Antioxidants from berries, citrus fruits, And cruciferous vegetables protect the pineal gland and enhance melatonin synthesis, which contributes to better sleep, immune regulation, and blood pressure stability. Furthermore, the anti-inflammatory and vasodilatory actions of these foods help regulate core body temperature and autonomic nervous system activity, two critical factors for achieving deep, restorative sleep. This ties directly into cardiovascular health, as poor sleep is an independent risk factor for clotting events and arterial plaque progression. Ultimately, a salicylate-rich diet, a quick fix, but a comprehensive lifestyle pattern that supports homeostasis in nearly every organ system. It gently nudges the body toward repair, balance, and resilience by providing the raw materials it needs to regulate inflammation, preserve vascular health, and maintain adaptive flexibility across changing environmental demands. This broad spectrum, systems-based approach reflects the elegance of human physiology, where healing is most effective when it aligns with the body's natural design rather than opposing it through isolated, high-dose pharmacologic intervention.